DNA and RNA. DNA, or deoxyribonucleic acid, the blueprint of life, is a complex molecule within the chromosomes of every cell that carries the genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. It's the hereditary material responsible for passing traits from parents to offspring. DNA is composed of two long strands that intertwine to form a double helix, resembling a twisted ladder. Each strand is a chain of chemical building blocks called nucleotides. A nucleotide consists of three parts: a deoxyribose, which is a 5-carbon sugar phosphate group, which is a molecule containing phosphorus and oxygen, and a nitrogenous base, which is a molecule containing nitrogen. There are 4 types of nitrogenous bases in DNA: adenine, A, thymine, T, guanine, G, and cytosine, C. The two strands of DNA are held together by weak hydrogen bonds between the nitrogenous bases to form double helix. Adenine always pairs with thymine and guanine always pairs with cytosine. The sides of the latter are formed by alternating sugar deoxyribose and phosphate molecules. The rungs of the latter are formed by the pairs of nitrogenous bases. Functions of DNA. DNA stores and transmits genetic information during cell division by replicating itself and transmitting a complete copy of the genetic information. DNA is passed from parents to offspring, determining inherited characteristics like eye color, hair color, and height. DNA plays a crucial role in various cellular processes, including gene expression, DNA repair, and cell differentiation. Now, we shall talk about RNA, ribonucleic acid. RNA is a nucleic acid composed of one single long chain of nucleotides, consisting of a nitrogenous base, a ribose sugar, and a phosphate group. DNA and RNA differs from each other in following ways. Firstly, each nucleotide of RNA contains ribose sugar with OH group at carbon number 2, whereas sugar in each nucleotide of DNA is deoxyribose, one oxygen removed from O group at carbon number 2. Secondly, DNA contains two polynucleotide chains, which are coiled round each other opposite. While the RNA consists of a single strand of ribonucleotides, which may be folded back on itself to form a double helix, DNA occurs in chromosomes, in the nuclei of the cells, and in much lesser amounts in mitochondria and chloroplasts. RNA is present in the nucleolus, in the ribosomes, in the cytosol, and in smaller amounts in other parts of the cell. The 4 primary nitrogenous bases in RNA are adenine A, guanine G, cytosine C, and uracil U. The sugar phosphate backbone of RNA forms through phosphodiester bonds between the 3-hydroxyl group of one ribose and the 5-phosphate group of the next ribose. Types and functions of RNA. There are several major types of RNA, each with distinct functions: messenger RNA, transfer RNA, ribosomal RNA, small nuclear, and microRNA. RNA that carries the genetic information transcribed from DNA to the ribosomes, where protein synthesis occurs, is called messenger RNA. It serves as a template for protein translation. tRNA molecules transport specific amino acids to the ribosomes during protein synthesis. They recognize the codons on mRNA and ensure the incorporation of correct amino acid into the growing polypeptide chain. Ribosomal RNA is a major component of ribosomes, the cellular machinery responsible for protein synthesis. It plays a crucial role in the catalytic activity of ribosomes. Small nuclear RNAs are involved in various cellular processes, including RNA splicing, telomere maintenance, and ribosome biogenesis. MicroRNAs regulate gene expression by binding to mRNA and inhibiting protein translation or promoting mRNA degradation.